the pandemic, like many companies around the world, AstraZeneca was figuring out how it could help in the fight against COVID-19. So we were, of course, making donations of millions of masks and other types of safety equipment. We were involved, the UK government, with COVID testing. Building on our RSV legacy, we started to develop long-acting monoclonal antibodies to protect immunocompromised persons who can't respond well to vaccines against severe forms of COVID. And we also initiated a partnership with Oxford. And it really all began with a phone call with John Bell, being the head of medicine at Oxford, calling our head of R&D, who then spoke to our CEO. And within a few days, there was agreement that the two companies would launch into this public-private partnership to develop a COVID-19 vaccine using Oxford's adenoviral vaccine platform. And the agreement was that it would be done as a nonprofit program during the pandemic. And AstraZeneca also further agreed that they would continue to offer the vaccine even post-pandemic at cost to any low or low middle income country. So that's what was done. And AstraZeneca at the time was not a key player in the vaccine space, so they really didn't have a lot of the internal infrastructure that was needed to carry this off. So I still feel amazed, that this was something that we were able to participate in. And the result of that partnership was over 6 million lives saved during the first year of the pandemic. More than 3 billion doses were distributed worldwide, which was more than any other vaccine brand. And that was to more than 180 countries. More than 2/3 of those doses went to low and low middle income countries. More than 473 million of those doses were delivered through COVAX to 130 countries. So this meant building supply partnerships with over 25 different partners. We also built the manufacturing capabilities of countries like Korea, Thailand, Brazil, et cetera, which will also have a long-lasting legacy for vaccine preparedness in the future. And then I think we all know this as well. I mean, it also still continues to blow my mind of how quickly this all happened. So within 104 days of that viral sequence being identified, the first in-human trials occurred, and then approvals for emergency use authorization 8 months later, and then a couple of months after that, the authorizations by the EU, the WHO, and then 1 billion doses being released for supply. And then key to this was AstraZeneca initiating at risk the manufacturing of those doses before any of the approvals happened. And that's how the release of so many doses was able to happen so quickly. So, of course, the key moment was this Oxford-led phase 3 trial, which was able to show a 70% efficacy of the vaccine against symptomatic COVID. But the trial was also run with a lot of questions. So there was different efficacy results depending on different regimens, and those data were difficult to interpret. And so from that point on, there was really a very linked interplay between clinical trial data and real-world data as different aspects of that needed to be understood. Everything from protection from severe diseases, the overall efficacy, older age groups, boosting doses, and of course, the non-ending saga of different variants of concern. And if you think about the trial, there was one specific thing that happened. The way that it was designed was that younger age adults were enrolled first, and then it built up to older age adults. And the result of that was that the final trial had actually very few data for older and elderly individuals, and so was not conclusive regarding the efficacy in those age groups. So that really led to this watershed moment in February 2021, when the first real-world evidence began release from the UK. And it was shown first with Scotland data and then with other confirmatory data like these from the University of Bristol, that the vaccine was in fact highly effective in older age individuals. After that, the US trial results were released in March. So this was the AstraZeneca run phase 3 trial. And this was almost a turning point, right? Because you had actually clinical trial now data, confirming what we saw in real world effectiveness data. And this showed, a high level of efficacy against overall, as well as in adults over the age of 65. With each variant coming out, real world evidence, of course, again, played a key role, showing continued high effectiveness against the different variants as they arose. And then finally, the question about what, in fact, though, was the benefit risk profile, which we've talked about already a lot today. But just here to say that, of course, this was a very important and critical evaluation that needed to be done, taking especially into account the differences in benefit and risk by age group, which led to most European countries limiting vaccination recommendations for this vaccine to older adults and elderly. But perhaps slightly less known is the fact that that distribution of the TTS cases actually really differed globally. So most of those cases were concentrated in the Nordic countries. You had a kind of a middle level with Australia, UK, and the EU, and then globally there was hardly any that were reported. Following that, WHO continued to support AstraZeneca Oxford vaccine as part of booster dose vaccination programs. So it was included in the WHO good practice statement as part of heterologous dosing recommendations. And so the vaccine did continue to be used globally, which led us to the ability to assess the effectiveness of booster doses, which was again shown to be at a similar and high level of effectiveness to mRNA vaccines in relation to severe disease. And then after the first booster dose vaccination, there became questions again, okay, first booster may be effective, but now what about the second booster? And at this point in time, our vaccine is really being used less and less globally. So there was only 2 countries worldwide that were able to produce any sort of data whatsoever if we get to effectiveness. So these data in Thailand, which was run independently from us as part of a community-based surveillance study, and then our own study of the Brazilian national surveillance data. One of the main points actually that I want to make here that I don't think we've made so far in exactly the same way is just to show that with effectiveness data, we have to be aware that a lot of those data are driven by the US data and also the United Kingdom, and in this case, Israel and Canada as well. So if you're a vaccine that doesn't have approval in the US, it means automatically you're not going to be generating the same level of effectiveness. So that was really impacting us and our ability to continually be able to show the effectiveness and the benefit of the vaccine. But I think another point here that I really want to make that hasn't been made so far, because we all know what the infrastructure and limitations could be outside of high-income countries in terms of healthcare structures for these types of studies. But it's also the fact that vaccines arrive in non-high-income countries later. Those vaccination programs are initiated later. They take longer to roll out. And this means that your effectiveness data is always going to be delayed if you don't have a big source of data from the US or similar countries. So as we look and we talk about these different subjects, but I think these are kind of the key things that we're talking about in these sessions, I would also just like us to keep in mind what can we do to make sure that there's equitable data from both high and lower income countries, and knowing that lower income countries need their own data that's applicable to their situation so that they can also make timely global decisions. And that's it.
We have time for one question.
Thank you. That was very interesting. I don't know if you can go back or just, I wanted to comment on your one slide showing the differences in risk of TTS in different areas of the world. And I'm wondering that could be due to reporting differences, but actually could be due to different genetic susceptibility in different areas. And I'm wondering whether you're looking at that in any way.
There has been research into that. There hasn't been any specific, I think very strong hypotheses or findings regarding that. I think there has been a little bit, but nothing conclusive. And I think this is something that can be debated very well. Yeah.
Thank you, Sylvia. Our next speaker, who has already spoken, is Steve Black. He's a physician, pediatric infectious disease specialist, more than 30 years experience conducting clinical trials, so he's not as young as he looks. And then he's led of
not as old as I look either.
I'm abusing my position, I have the microphone. So 25 country global vaccine data network, of which he is going to speak.
Okay, thank you, Hector, and thank you for the chance to talk briefly. So just a word about the genesis of the Global Vaccine Data Network. This actually arose after the narcolepsy outbreak in Europe following the H1N1 2019 influenza vaccine. And at that time, if you remember, which many of the audience will, different countries found very different levels of risk, varying from a 16-fold increased risk in Finland to no increased risk in the Netherlands. And then the US CDC funded the Somnia study, for which I was the PI, but that took, because the infrastructure didn't exist, took a long time to provide study results. So a group of us felt that maybe we could do better with this, and founded the Global Vaccine Data Network. The problem that the Global Vaccine Data Network is trying to address is that single country studies are often limited to one or two vaccines. We've seen the real world evidence reported on today really has been limited pretty much to two RNA vaccines and the J&J vaccines. Also, rare events require very large populations to assess them, and hence multi-country collaborations are useful. And as Bob Chen has pointed out, mixed schedules and complicated schedules increase the need for this, because for most outcomes you want to stratify by brand, by dose number, by age, by gender, and that leads up to 36 different strata. So even with VSD with 12 million people, you start dividing that up and you only have about 1/3 of a million per strata, which limits your power even in that large study. So the global network is a network of sites agreeing to conduct coordinated studies using single protocols for active surveillance and epidemiologic vaccine studies that are multinational and, importantly, investigator-led. And in 2021, we received funding from the US CDC to assess the safety of COVID vaccines in this network. So a word about the network. There are 34 countries with more than 400 million people involved potentially to analyze, and they're quite variable in terms of capacity, as I'll discuss, and in population. Some are academic centers and some are government centers. For example, Nick Andrews and Julie Stowe from the UK here. So and we also have participation from the Alive Network in Africa that Claire will talk about following me if I survive this talk. And also we collaborate with the VAC4EU network that Miriam spoke to you about. The COVID experience so far is these are the projects that we have undertaken, varying from background rates, which I'll show you examples of, to outcome or association studies for myocarditis, VITT, vaccine-mediated enhanced disease, which we've not seen data for yet, and also genomics and maternal immunization studies as well. And I'll talk to you a little bit about some of these in a second. We have a dashboard which we display results, and you can go online to the GVDN website, and there's a link there, and you can click which country you want to look at, which outcome, and then look at the background rates over time. This is for Finland, for ADEM over the study period, for example. And the background rates data have been published in Vaccine. We also have an observed expected dashboard. This is for a meta-analysis of all the sites. And you can see here the point estimates and also the confidence intervals around the estimates for myocarditis. And this data is what you'd expect is associated with DNA, with RNA vaccines. So the current status is that the background rates have been published. The observed versus expected dashboard is active, but we're waiting for Anders Hvid in Denmark, who's leading developing a paper for that. And when that's published, then that'll be publicly accessible as well. And the association studies are in progress as well. We're collecting specimens for genomic studies of myocarditis, pericarditis, GBS, and VITT. And I'll tell you a little bit more about that in a second. We're going to have a meeting in April of next year when we share results for almost all of these studies. So a word about genomics. Genomic studies of drugs have revolutionized drug therapy, allowing for personalized therapy for many drugs. But identifying genetic markers for vaccine adverse events has not really largely been investigated, mostly because many of these events are rare. So you need really a large population to identify cases to study. But potentially they could facilitate better understanding of the pathophysiology of the events, potentially allow for personalized vaccine schedules as well. If you knew who was at risk for myocarditis, you might give them a vaccine with a different platform, etc. Actually, I should say Bruce Carlton is at the University of British Columbia in Vancouver, leads the genomics effort. He's a well-recognized investigator in terms of genomic studies for drugs. And he's going to identify 300 cases or more for each of these rare outcomes. So that's why the global network is useful. The ALIVE project, I'm not going to say anything about except that we've collaborated with Claire, who will be speaking next, to conduct vaccine safety studies in 8 countries in Africa. So in terms of lessons learned, the GVDN COVID vaccine safety studies were funded 1 year after the start of the pandemic. So not having infrastructure in place before the pandemic made for a slow start for us, because we had no existing relationships. We all knew each other, but had no contracts in place, no ethics approvals in place. Some sites took more than a year to develop, to get ethics approval. We didn't have protocols in place or statistical analysis plans. You get the picture. We had nothing in place. So that took us a long time to get off the ground. The other thing we did at the beginning is we had sort of view the network as an open tent. So when we were developing protocols, a lot of people came into comment who were in no way ever going to contribute to the study, and that while they contributed a lot of interesting ideas, it basically slowed us down and led us astray in some ways, because they were not really aware of what the capabilities were of a site, because they weren't really going to be a contributing site themselves. And I think, as you'll see with Claire's presentation, inclusion of low-income countries, not only in Africa, but in India as well, has required developing a different data collection system. So while we're successfully conducting investigator-led studies, timelines for the projects have been long. We're developing a rapid response protocol to address new safety concerns. And we need to develop decentralized infrastructure, basically as Miriam described to you for VaxVU. So to have it be decentralized, but at the same time not fragment the network. We don't want everyone to go off on their own, but at the same time, we'd like to foster more independence. And as always, for vaccine safety, obtaining sustainable funding is a challenge. Some sites have no or very limited electronic healthcare data. Argentina is one, South Africa and the Alive Network are another, India is another. Some sites have inpatient data only. Other sites have everything you can imagine from lab and radiology data. Time delays to access data vary from near real time to 6-month delays. So we're trying to see this variability as a strength rather than something to be worried about and try and build on it as we design different studies. So the strengths of the GVDN, to sum up, is it is a globally coordinated network with 30 plus countries, and the number is growing. Indonesia has just joined, for example, and Korea just joined just before them. We now have existing administrative infrastructure and agreements in place, so we could now respond to a future pandemic more quickly. We have global experts who are capable of leading new studies. And diverse vaccine exposures and diverse populations, including subpopulations of pregnant women, children, and HIV-infected individuals, are a resource that can be used for future studies. As I said, now with inclusion of Indonesia, Korea, and soon Brazil, this population will increase to more than 650 million. So I think I'll stop there. You can read the last 4 bullets since the timer just zapped me out. So I'll stop here.
Thank you.
And answer any questions. Thank you.
We have time for one short question, so there is sufficient time for the answer, please.
I'll try again. Shorter. Oop.
Oh, thanks, Bob. Gabrielle Brueggemans.
You can try again later, Bob. Yeah.
Seppi. Thanks, Steve, for a clear presentation. One question. You indicated that often certain capabilities were not available, particularly in low resource setting. There might have been differences in different settings, but was there a sort of a core capability that was often not available that we maybe should capitalize on from your perspective?
Are you saying he's taller than me?
Yeah, I think as I said, we're trying not to throw people out, if you will. We would rather look for different pathways to collect data and foster the ability to analyze and interpret data at these sites. One thing that has been challenging, for example, something as simple as background rates. For a background rate, you need a denominator, classically. So we've been trying to work with statisticians to develop models to basically built on the successive disproportionality models for passive reporting and apply that to actively collected data to look for basically a comparison that you could use if a vaccine is introduced. So I think rather than people ask, well, how did you pick this group of people? We didn't pick them. If people are interested, we're interested in figuring out a way to have them collaborate. So do you want to let Bob ask his question?
Afterwards.
Okay,
one question
over beer later, Bob.
Thank you. You have opportunity afterwards, Bob. Our next speaker is Claire Cotland. She is a BSc, MBBCH, DCH, PhD. She is scientific coordinator of the WITS Alive Consortium. And although some people don't know that she has more than 18 years experience in clinical research in vaccinology. Please.
Thanks, Hector. And thank you very much for inviting me to present today. So I'm going to give a bit of an update, some background information on an active COVID-19 vaccine safety surveillance projects that we set up in Africa. So, as Steve has already mentioned, there are some projects that were established. So I'm going to give you the background on two projects that were done. I'm from South Africa. One of the projects was done in South Africa, but South Africa is a middle-income country and therefore not Gavi eligible. So of the two projects that I'm going to tell you about, the one is called the ACVAS study, African COVID-19 Vaccine Safety Surveillance Study, which is demonstrated on the left. And this is a collaboration where we tried to establish active COVID vaccine safety surveillance in 8 AMC 92 Gavi eligible countries. This project was funded by Gavi, and GVDN was our technical partner. Shabir Mahdi, who's my direct line manager, is the principal investigator on the study. The second study was done in South Africa, and this was funded through the GVDN network with CDC funding that Steve has already mentioned, and that was conducted at 5 sites across South Africa. Across the 8 countries that we tried to establish vaccine safety surveillance in across Africa, data collection was not initiated in one country because of delays in getting ethics approval and the site established, but in the other 7 countries we had 48 sites. So the aim of the study was to set up a system of hospital case-based surveillance of a predetermined list of AESIs following COVID-19 vaccination. And the primary objective was to estimate the risk of these predefined AESIs with an acute onset in a set period following vaccination using a self-controlled risk interval study design. The secondary objective was to contribute data to the GVDN network that Steve has already mentioned with the association studies. So this is a list of the ASIs that we were looking at specifically, and it's from the Tier 1 list in several different documentation. So with the study flow, the protocols were developed mainly by the investigators and were aligned with the WHO template for safety surveillance. The way that the studies were conducted was that when patients were admitted to hospital in these countries, they were identified by in-country investigators and investigational teams. They were added to a hospital admission log, and then the study staff reviewed that log and identified patients who were possibly admitted with an ASI. Those patients were added to a screening log, and they were then approached directly by the study teams and invited to participate in the trial, in the study, sorry. So there were some screened participants who didn't participate, and if they were eligible and required and happy to participate, they were consented, and then they were enrolled. There were a couple of forms, patient demographic form and a clinical history form that were completed at that point. And then depending on the condition with which they were admitted, there were different forms that were completed. The patients were then followed up to discharge, and discharge or outcome data were collected. For the ACVAS study, the grant was finalized in October 21. The first patients were enrolled in April 22, and patient enrollment was ended in March 23. So we're in the phase of data analysis at the moment and writing up reports and results. The South African study was initiated a bit earlier because the grant was finalized earlier, but the sites started at different times between October 21 and November 22. Again, data collection continued until March 23. So training slides were prepared and recorded by Wits Alive, Wits Vida, and GVD and teams in English. The PowerPoint presentations were made available to the study teams that they could either present to their teams in person. So the photos on the right of your screen is the Ethiopian team's investigator training his teams. Some of the slides needed to be translated, and they could be translated obviously by the investigators and site. The training included protocol overviews, study procedures, case definitions, levels of certainty, and according to the Brighton collaboration definitions. And then also data capturing onto the REDCap database. Data collection instruments were designed initially in a Word document and were available in Word document and PDF. Some sites needed to collect data on paper, so they could be printed off that as required. And the database was developed in REDCap by the Alive and GVD and teams, and that was made available across all of the different sites. Personal identifiers were not included in the database. We did develop an Excel spreadsheet which the individual studies, countries, investigators held, and individual patient identifiers, which were obviously essential to be able to trace results, laboratory results, etc., were maintained on site. They did not cross country borders. They were not put onto the database. The database was hosted at University of Witwatersrand, based in Johannesburg. We also had pre-calculated levels of certainty inbuilt into the REDCap database instrument. The data could be collected either using the online interface on computers or on the REDCap mobile app, which was very useful, particularly out in the field, and that could be done either online or offline. One of the challenges that arose when data were collected offline is when the tablets were brought back into the coordinating center on site and the data were uploaded, there were some synchronization issues, and sometimes patients were allocated the same numbers. So that had to be manually corrected by the Alive epidemiologist working with the site teams. We wanted to make sure that all of the documentation were available to the different sites. So this included obviously the protocol, the different formats, the stats analysis plan, so any document that would be included in an investigator site file. SharePoint and other things are not always available or accessible by other institutions, and we used OSF, which we had set up. So we set up different projects in OSF, and we were able to upload documentation and share this with the study sites. The sites were also able to upload their ethics approvals and training records onto this OSF platform for us. So as a summary of our initial results, this is just a flowchart of the screening and enrollments across the 8 African countries. So in total, there were over 60,000 patients who were screened across the 8 countries, so including South Africa and then the 7 ACVAS study countries. And there were 12,700 patients who were enrolled on the study, 6,795 across the low-income countries settings and 5,961 in South Africa. For more data, you're going to have to keep looking out for some publications that will be coming out over the next few months. We're just finalizing the different analyses at the moment, so they will be coming out soon. One of the big issues and one of the lessons learned is about the access to vaccinations, source vaccination data. So if I just look at the ACVAS study data, there were, as I've mentioned, 6,795 patients were enrolled. And of those, only 32%, almost 33%, were vaccinated with a COVID-19 vaccine. The source of the vaccination data was only known for just over half of those. And of those, the vaccination card was accessible. So it's a paper-based vaccination card. The vaccination card was available to the investigators for half of those. Quite a few of them were self-reported by the patient. Some of them were reported in the medical records, and only 2.5% were recorded on a digital platform. So when we look at the vaccination data, we do know that there were vaccinated patients with GBS, myocarditis, pericarditis, and TTS. And if we just look at the number of forms that were collected, if we look at GBS, for example, 86 forms were collected, but only 15 of them had LOC 1, 2, or 3 data available, and only 4 of those had vaccination data. Okay. So just from the lessons learned, funding and contracting is challenging, time-consuming. Reimbursive grants are very challenging in LMICs and need to be avoided where possible. Ethics approval, this was run as a study, so ethics approval took anywhere between 7 and 120 days, and it did significantly impact the time to set up the studies and roll it out. Database development, we did have 2 databases that we developed. We had to update a database, and this has posed some challenges just with merging the data and with the analysis side of the data. And REDCap on the whole was great. I have mentioned the cons of this synchronization data. Vaccination data access and validation was very challenging, and mainly because it was there was only an electronic data system available in South Africa, which was EVDS, but even that there was limited access to, and otherwise it was very challenging to get the paper-based records. Also, one of the challenges for the monitoring team in Ethiopia was that they use a Gregorian calendar, which makes all of us 5 years younger than what we are, but the Ethiopians could convert their dates easily. So that is basically, monitoring was a challenge as well, and staff and training did pose some challenges with different levels of staff being used in different facilities in different countries. And there were some language barriers which did pose some challenges in some of the countries. REDCap has introduced a translation facility in the database, which could get around some of those problems. So thank you so much.
Thank you. We have time for one question. Well, I will ask the question then. During the flu pandemic and afterwards, we did with Steve Black, Miriam Sturkenboom, Caitlin Dodd, Sylvia Perez-Vilar, and others, studies of the use of self-control case series in vaccine safety, collaborative international projects. Did those studies contribute in any way, shape, or form, or you started from scratch again?
Sorry, I don't understand, did they contribute to us making the decision to use the SCRI, or did they?
Or in the design, or in any other shape, or form, or way?
Steve, should I bounce this back to you?
Well, I think that's an answer by itself. Thank you.
Sure.
Any other question? Anybody? There is time for one question more. Thank you. Thanks. There is some time for anybody to ask a couple of quick questions for anybody among the presenters. Bob Chen is the first one, and then somebody else can ask another question.
Yeah, so Steve, my question was that obviously sustaining these networks are challenging, and part of it is that the vaccine is only part of the pharmaceutical product market. Has anybody in the drug safety domain approached GVDN to see if that network could be used? Because many of the countries are high-income countries with probably automated pharmacies, et cetera.
Yeah, actually, there's someone named Jeff Peterson in New Zealand who's a drug safety person who's very interested in beginning discussions in that with that type of thing. Most of the all of the investigators within GVDN are primarily interested in vaccine safety. The other area people have talked about is using this for vaccine effectiveness or risk-benefit assessment, but really that isn't and that came up 10 years ago at the initial meeting of GVDN, because it was initially GVSDN with only safety, and the S was taken out to allow inclusion of a broader scope, but that's not gone anywhere yet at this point. Does that answer your question? Or? Yeah. Okay.
Thank you. There is time for one more question for any of the speakers. Anybody? Going one? Going twice? Please. Please.
Yes, question for Steve, really. I mean, the rationale of having lots of participants and getting the data quickly, there's often power issues and you get heterogeneity, and you've got either a few definitive studies adequately powered in a number of settings, or you have joint data inadequately powered in any one setting. Do you see that tension, and how do you overcome it?
That's actually the tension we're trying to overcome, essentially. I mean, it's much easier and much quicker to do studies in one country. But if you look at the genomics, for example, assembling 300 cases of Guillain-Barré syndrome in one country is not possible. There is no way to do that. And the other thing about heterogeneity, it's different populations, and that's one of the reasons I asked about the AstraZeneca question, to see whether that's real or not real. And really the only way you can answer that is with a global network using a common protocol to look at that. So yes, heterogeneity is an issue in terms of speed, and it also poses analytic issues, but it also, because of diversity of population and exposure, et cetera, allows you to look at greater, look more broadly as well. And I think in one of the lessons that Claire didn't talk about in terms of the ALIVE study is none of those countries individually would have the power to do a study for these rare outcomes alone. And in fact, even collectively, the power is very limited, but they can begin to contribute to global studies, which allows them experience of participating and being able to address safety concerns within their own country from a data source that the majority of which may not be theirs, but at least they contributed to it and can speak from that perspective. So there's trade-offs, but it's complex.
Thank you. Thanks, everybody. This session is closed.